gonna be it.
Yeah.
But there were other things that I learned that, I actually learned. And you kind of hedge your bets, right?
Yeah.
Hedge your bets.
Also, what I can't find what I want to do online. That's the other problem. Or I can for an exasperating
Exorbitant
relating amount of
exorbitantlating.
Like, that number's so big, I'm excited in a bad way.
I don't even know how to contemplate that large number.
And I feel like if I gave somebody that much money, they should just give me a degree, whether I've shown up to class or not. Like, they owe me.
That's what Ivy League schools are about.
Oh, shots fired.
Wait, what?
Boom, snap.
What?
You missed the shade that Kiki threw. It's like dark in here. There's so much shade everywhere.
I missed it. Can you repeat?
Nope, no more. Nope, that was a one-time only.
I'll have to rewatch the entire episode from beginning to. Oh well, thankfully it's the beginning. Okay. Hey, we're live. Is there a chat room? There
is a chat room. There is a chat room. There is a chat room. Our chat room at twist.org/live. And there's also a wonderful chat room on our YouTube channel. Hey, everybody, we're here. It's time for some science. Yeah, science time, right? Are we ready to go?
Mm-hmm.
Yeah, ready to start this thing? Okay. The bat signal has gone out. The science signal has been sent from the science city of Gotham. Starting in 3, 2. This is TWIS. This Week in Science, episode number 689, recorded on Wednesday, September 26th, 2018. The Scientific Sense. Hey everyone, I am Dr. Kiki, and tonight on This Week in Science, we are going to fill your heads with a gut feeling, a sickly smell, and Japanese robots. But first...
Disclaimer. Chances are just that. Things that happen by chance. The unexpected meeting between two people that otherwise would never have happened. They never would have met if not for circumstances in which unintentionally they do meet. They talk, and chances are that's all they'll do. Talk for a few passing moments, enjoy each other's company, and go their separate ways. But there's also a smaller chance that the conversation leaves a lasting impression on one of them. And though the probability becomes slimmer still, there's a chance that the conversation leaves a lasting impression on both of them. So much so that they decide that the conversation, the one they just happened upon by chance, should continue. That, in fact, it should never end. Science is a lot like that. Conversations started long ago, often by chance, that no one has tired of having because it's just too damn interesting to walk away from. Just like This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again. We do this periodically, once a week. Come to the internet and discuss science. Been a wonderful week. I was trying to avoid the internet for most of it. Ha! But now we're back. We've got science to discuss. What did I bring this week? I have stories about a new sensory system, dead mosquitoes, and drug addiction. What do you have, Justin?
Oh my gosh, what have I got? I have got brutal people of the past, microbial trash cans, and what is my other story? And I have another story, but I've completely
a mystery.
yes. A story about your nose.
What? Did anyone bring the Japanese robots?
No.
We're going to have to talk about the Japanese robots. We'll talk about that. And Blair, what is in the animal corner?
Well, speaking of the nose, I brought smelly sicknesses, and I brought some animals that just don't know what they're supposed to eat.
How do you not know what you're supposed to eat?
They're breaking all the rules. We're going to talk about it. They don't know what they're supposed to do, according to science. They're not listening.
They're not listening. Well, everyone, I hope you are listening up because it's time to jump into the science. But before we do, I would like to remind you that if you have not yet subscribed to This Week in Science, you can do so. You can find information at twis.org, TWIS.org, about how you can go about doing that. Or you can look for This Week in Science or TWIS in all the good places that podcasts are found. Okay, let's jump on in to the genie in the lab. I dream of genie. Can we make a wish? Are we going to make a wish here? A wish for a
endless funding. Oh,
yeah, that's a scientific wish for sure. The magical wish for no more mosquitoes. That people,
but they're pollinators.
I know we've talked about the benefits of mosquitoes before, but there are many, many downsides, especially as we discuss emerging diseases, diseases that are beginning to affect the more tropical-ish portions of the United States as climate change spreads warmer and wetter environmental conditions further northward. We are also seeing a spread of disease-carrying mosquitoes. And, it doesn't really matter. Just the whole world. We are concerned about diseases spread by mosquitoes, right? We've talked previously on the show about gene drives. Do either of you remember what I've said about gene drives in the past? What
Too
they
dangerous.
do?
Don't do it.
They can drive off the road.
Well, yeah, maybe they could drive a species off the road. So
horizontal gene transfer. What if the entire biome collapses?
Right, but let me define what the gene drive is first for people who may not have heard this discussion before. Gene drive is a manipulation of genes, so inserting a particular gene or modifying an existing gene so that it is so dominant, it blocks all other variants of the gene. It drives itself into the population.
Ultra dominant.
Ultra dominant. That's right. And depending on the trait that is carried by the gene or that gets expressed when the gene gets expressed, it could have differing effects at the population level, especially if it is a gene involved in reproduction. Right? So researchers have been talking about using gene drive to reduce the reproductive abilities of mosquitoes. And they have talked about taking gene-modified, gene-edited mosquitoes and releasing them into the wild. And this is what the point that Justin brings up. It's like, what happens? How could it, what if it spread to other populations? What if a gene escaped the population? When you leave the lab, you leave the controls, and you don't know where the genes may end up. So these are the dangers of gene drive. But let me tell you where we are with the research. A study published this last week in Nature Biotechnology. This is big. This is big, everyone. Researchers used CRISPR Cas9 targeted disruption for an intron 4 exon 5 boundary that was aimed at blocking the formation of a functional gene called AGD-SXF. And when this gene was manipulated in males, didn't change anything. Males still very able to reproduce, go on and reproduce. However, females that were homozygous, which means that they have 2 of this particular disrupted gene instead of just 1 copy. They have 2 copies, different chromosomes. When they had 2 copies of this disrupted gene variant, they showed an intersex phenotype that is sterile, unable to reproduce, and the gene drive reached 100% prevalence within the laboratory population within 7 to 11 generations.
That's
It
very aggressive.
It reduced.
Now, I don't know how long mosquitoes live, how long it takes to produce that many generations, but I feel like it's probably pretty quick. I feel like that would happen within a month or 2.
Right, exactly. Justin, please get a little closer to your microphone.
Oh, sorry.
There we go. Fantastic. Exactly. And what if you're having continually reducing egg production? That means nothing for the sperm gametes to combine with. And in the laboratory, this is not in the wild, they reached total population collapse. The entire population in the laboratory of mosquitoes collapsed. There was to a point where there was no coming back. The mosquitoes they died out in the lab.
This is what's so scary, that it's so effective, that if there was some sort of unexpected horizontal gene transfer, you could lose all of another flying insect. I don't know.
Well, here's
maybe not
upside of it, though. Here's the upside of how quickly it operated. Horizontal gene transfer, you might need many years of chance and probability for this to even possibly come up, let alone stick, let alone be the right gene that transferred and all the. Right, so
you're right. That's true.
The fact that it's so effective, the fact that
it might work quick enough that it doesn't spread.
Put it out there and we'll leave it there for 50 years and see what happens. No, I mean, this sounds like it could be over and done with whatever population acquired this very quickly.
Kiki,
very quickly.
It's species specific, though, right? For the mosquito.
This is a sequence that is highly conserved in the Anopheles mosquitoes that have been analyzed. So it is specific to the genus.
Okay.
Now mosquitoes,
not specifically species, and there are subspecies as well. So this could, the mosquitoes in areas where various populations and species or subspecies types overlap, there may be the possibility of reproduction between the different populations that could lead to the spread if this were,
so
released into the wild.
This before, though, in nature, wasn't there a gene, like a sort of a gene drive type or a sterility type encounter between mosquitoes? This is the North American mosquitoes were completely replaced at one point with South American mosquitoes, because whenever they interacted and mated, the North American mosquito became sterile as a result, and nothing, and no effect on the other mosquito. And over time, this buildup of sterility really pretty much almost entirely wiped out the population of one type of mosquito in favor of another. So we've seen something like this in nature.
Yeah.
Right. The issue here, though, is that it's the 100% spread in the population. There were resistant variants that arose through natural mutations within the population, but none of them blocked the spread of the drive. None of them spread they did not block the functioning of the drive to decrease fecundity.
So I've always defended all life forms on this earth, save one, which has been the lily mosquito, the much maligned, the bloodsucker, the pest. However, after our recent knowledge gained of knowing that they are pollinators, I need to know how big of a deal these pollinators are. Because if it's just a little bit of pollination, I might be like, okay, we'll figure it out. We just need more bees. But if they're
easier said than done.
Yeah.
Right. If they're plant specific, that
Right.
Yeah.
pollinators aren't pollinating. If there's certain, you start taking out pieces of the big ecological Jenga game, and things get shakier.
Yeah, absolutely. Yeah, I think that's really the question here is, one, can we make this a little more specific? Because I did just look it up. Anopheles is about 460 species of mosquitoes. About 100 of them transmit malaria. So that is that's it's only a quarter. So that's kind of scary if it's going to affect this wider range, right? So I would like to see it be more species specific, and also, see a little more research about the impact of potentially removing these guys from an ecosystem.
'Cause there's not coming back
If
from
the benefit outweighs the cost.
Yeah,
Right.
but there's
Still
not a
something to
coming back
consider.
from this. It's not like, the one thing that normally would take place if this was a pesticide or something of this nature. We'll try it over here in an
Yeah.
outdoor controlled environment. Or maybe one country will raise their hand and say, like, it's such a problem here that we're willing to take the chance that there's a repercussion. And then if there is, it's local
Mhm.
and it hasn't affected the planet, right? Like it doesn't, it's not pandemic-y. This sounds like we either have to pull the lever or not, and there's no going back. There is
Once it's
no
let go. Yeah. There's no returning once we release it into the wild. And so the question is also, this is a laboratory situation where these mosquitoes, they're held in containers with no competition. They've got plenty of food. There are no predators. There's no other measure of fitness for these animals,
whether they're
insects.
alive or not.
What is it? Are you alive or are you dead? Are you reproducing? Are you not? That's it. But whereas there could be a reduction in the female's ability to survive, to reproduce in the wild, that wasn't elucidated by this study in the laboratory. So although it reached 100% of the population in the laboratory, that doesn't mean that it would have the same kind of effect in the wild. If these females are less able to survive, they're not going to spread it as easily. However, the researchers do say in their discussion that an additional reduction in fitness over the experimentally observed value of up to 40% would still allow the drive to reach 100% frequency and cause population suppression, although it would happen more slowly. It would be a slow burn instead of a rapid fire effect. And so there are potential controls on this, and in the wild, with aspects of natural influence on the genome and selection pressures, maybe it would affect the drive moving into the population in a different way. And so we don't know it until there is a wider, more natural experiment, and by natural, I mean in a natural setting where there are all of this survival and selection pressures that mosquitoes would experience.
Mhm.
But how do you do that without actually letting it go?
Yeah,
This is
that's the question.
Yeah, so it's a very interesting question that researchers are pushing forward on this research. Obviously, the ability is growing in targeting reproductive genes and having an effect on mosquito reproduction. And so we need to really start having a conversation about what we think about the actual use of these technologies and what kind of evidence is necessary before we actually allow it to be released into the wild.
Mhm.
Big conversations people need to start having. Another cool conversation, if you're, maybe right now talking about these mosquitoes, you're getting this gut feeling of, oh, this is great. Or your other gut feeling is, I hate being bitten by mosquitoes, and you're feeling all icky and go away, mosquitoes. And your gut feeling is gene drives are awesome, right? You could be having this amazing gut feeling. And there really is feeling in your gut. Did you know this?
What?
What? Yeah. So, a couple of years back, researchers realized that there's a lot more going on in the intestines in terms of sensory input than we have previously understood. And they looked at let me see if I can find my link. My links are not opening. There you are. And they looked in mice at the small intestine, where they were able to see how quickly signals start to be received and sent out. And they found in the lining of the gut, in the small intestine, there are all these little, finger-like projections in the small intestine that increase the surface area and allow you to absorb things. But they also, these little finger-like appendages, they have numerous epithelial cell types. These are different cell types that sense the luminal, the interior environment of your gut. What's it like in there? How does it feel in there? And some of these cells they have discovered are they trigger action potentials. They actually trigger an electrochemical potential across their membrane. And in studies that they have recently done, they have directly connected the presence of sugar in the gut to the activation of these, what they're calling neuropod cells, that stimulate through sensory, just like your sense of touch in your skin.
Yes. So this
just
is
like
what you're
the sense of
describing.
touch. But these neuropod cells synapse with vagal nerve projections, sending messages within seconds from your gut through the vagal nerve to your brainstem. And it's through a glutamate pathway. So it's using neurotransmitters.
So you're
Previously,
telling me
yes,
that within my intestines,
there is
I
sixth
have
sense.
millions and millions of little fingers
Yes.
touching my digested stages of food as they pass by.
Yes. Feeding
And
the
it's
food.
somewhere in my brain, I am paying attention to the feeling of my digested food as it passes through.
Yes. Yeah. So researchers historically, they were like, okay, so the gut, we found all these hormone signaling systems where guts release things like cholecystokinin and other hormone-based compounds that move very slowly. And the information is passed from our gut to our brain within, this is why researchers say this is why you feel full within 20 to 30 minutes of starting to eat. Because the leptin and the cholecystokinin and these hormones that are released in your gut during the process of digestion, they take a long time to get to your brain because they're traveling through your blood. And they have to get all the way up to your brain and tell your brain what's going on. And it's a long time to travel from your gut to your brain if you're a hormone. And researchers have been like, but we know it's got to happen faster than this. But before now, they have never found the synapses. They have never found the cells that talk to the neurons that connect to the brain. This is the first time. And now you can basically say the sixth sense, it's in your gut. You do have a gut feeling. You have a feeling gut.
It's like touch taste. It's
yeah.
To taste.
So there is, though, this is curious. This is so there needs to be, and I think I've remarked about this and joked about this in some way that we humans aren't in control of our brains. Our brains tell us that we're hungry by sending messages in pangs in our guts that what is this? We don't just logic and reason our way to it's been a long time since lunch. I should probably put nutrients that I need to survive into my body now. No, we might feel a hunger pang.
Mm-hmm.
The fact that there's a mechanism feeling to see if we've put anything in there and going, hey, the human hasn't put anything in us recently. We better go have a discussion because I'm feeling nothing down here right now. We need to fill this up.
There's no sugar in here. Yeah. There's no fat in here. There's no protein in here. Whatever the neuropod cell is reacting to, right? It's not like if it's a hormonally based system, it's not sending a message from 30 minutes ago. Hey, the stomach was empty 30 minutes ago. No, your stomach is saying, I'm empty now. I'm hungry now. Fill me up. Or it is saying, I'm full. There's a lot of sugar in here. So, hey, let's get the brain to tell the gut to do some different digestive movements and to increase peristalsis or to increase the release of other certain compounds to aid in digestion. So there's a much more rapid communication that is going on than we've ever really understood previously. And with the whole concept we've discussed about the microbes in our gut, the microbes are potentially communicating with our brain in a rapid fashion through these channels as well. Dun dun dun. Yeah, so there's some exciting stuff.
So that's the other thing
Feeling
too.
gut.
That like maybe one of these, like, this is pure speculation-y things, but no, it's stimulus response a little bit when you get used to a stimulus. So if sugars are triggering this, is there a desire to consume sugar so that the feeling is restored, the touch that sort of like people maybe who tap their fingers a lot, or getting a maybe it's like getting a massage to the intestines to have something to interact with.
I'd like my sugar massage, please. Yeah, so the researchers, this researcher Boraquez from Duke University, and his team are looking at how this sense might be involved in figuring out the types of nutrients, caloric value, and all sorts of aspects of nutritional aspects of the foods that we eat. Does our sixth sense suss these things out? There's a lot of sibilants in this. I apologize. Is it sussable? That's right. The gut sense. Feelings. And I'm feeling excited because JAXA launched a sporty little mission, according to the Japanese aerospace agency. They're like, yeah, NASA, you create the big, strong, solid devices. We make sporty ones. So JAXA launched Hayabusa 2 recently, and Hayabusa 2 rendezvoused with an asteroid known as Ryugu. And this asteroid, it's a lovely little asteroid, according to pictures sent back by the spacecraft, which is it's a very rocky place. So there were 2 landers that rendezvoused. The Hayabusa craft came within a couple hundred meters of the surface of Ryugu and then dropped its landers. There are a couple of rovers, actually. One of them they were having some issues with, but a couple of rovers on the surface of an asteroid. And this is as Hayabusa 1's mission before it and a sample return mission. They are spending some time on the asteroid, and then they will be coming back to Earth, bringing stuff back.
I thought Hayabusa 2 was gonna.
Are they coming back?
No, I thought it was gonna dive on into it. But maybe I miss.
No, they didn't dive into it. They are.
No, they've got
on it.
I thought they had they've got the landers and the rovers there, but I thought before the mission was over that Hayabusa 2 itself was going to crash it.
Am I wrong then? But
I
they're
don't
on.
know.
Okay,
I don't know.
We're on an asteroid in space. Japan's
That's
got
awesome.
little robots on an asteroid, and they're cute little rovers.
Hayabusa 1 was close to landing on the asteroid, but it turned out that the asteroid Itokawa was a giant gravel asteroid. It wasn't as like nice hard landing spot kind of thing that it was looking for, and it got really close and then kind of fell off. But this is cool. That was a pretty neat photo that you had up there too. I suggest anybody who's just listening, go look for the Hayabusa 2 photos.
Yeah, on Twitter and on Instagram, Hayabusa2 at JAXA, I believe, or at least Hayabusa2 you can look for. And the rovers, they're not wheeled rovers like the ones we have on Mars driving around. They're hoppers. They hop. They hop some 50 because there's not a lot of gravity on the asteroid. Whenever they take a hop, they get some 50 meters above the surface of the asteroid.
Wow.
Giant
That sounds so
grasshopper
fun.
hops.
God.
Yeah.
That's-
And they're according to this article, Hayabusa 2 still has a pair of rovers on board that it will also deploy at a later date. There is a sensor-packed cube that will be delivered to the surface, and there you go. The Hayabusa 2 craft itself will eventually land on the asteroid, take a sample of material, and come back to Earth. It's coming back, but the rovers are not. These landers will be there. We're gonna
Just
leave
leaving our trash.
That's right. Or it's not trash. It's our little robot toys, our robot friends. We're gonna have friends all over the solar system.
We're just like a toddler in the backseat of a car, just throwing toys out the window as we go.
It's a great analogy. Oh my goodness. This is This Week in Science. Hey Justin, what did you bring?
Let's see. What have I got here queued up for the
There.
yes. A little bit of history here. When the saintly Spanish fleet arrived in South America, the kindly conquistadors described the many brutal activities of the native peoples. These included things like torturing of children for discipline. There was butchering and decapitating. There was mass ritual cannibalism. A bloody, savage people who could really benefit from a brutal, tyrannical conquering and looting. That's if you believe everything the conquistadors said in their version of the events. And as it happens, there isn't a other side of that story to rely on. That's how brutal and complete the oppression started and lasted for hundreds and hundreds of years. So, 1951, archaeologist Samuel K. Lothrop excavated 220 bodies at a 1,500-year-old site in Panama's Playa Venado region. Came to no surprise that he found them buried alive, butchered, decapitated, hacked, mutilated, killed.
Yeah.
Physical evidence from a time, what is that, 680? From a time far before the conquistadors, confirming what we already knew from the conquistadors' accounts. Only that after taking a fresh look, it looks like he got it completely wrong. New evaluation of the site remains by Smithsonian archaeologists revealed no signs of trauma at or near the time of death for the 220 individuals. No evidence of ritual killing. According to Nicole Smith Guzman, postdoctoral fellow at Smithsonian Tropical Research Institute. Lothrop's work was back in, he did the excavation in 1951, wrote it up in 1954. It was likely, they say, a result of underdeveloped 1950s methods for mortuary studies and literal reading of the Spanish accounts of indigenous peoples as part of his research. Aha! This is quotey voice of Smith Guzman. We now realize that many of these Spanish chroniclers were motivated to show the indigenous populations they encountered as uncivilized and in need of conquering. Adding that many of the accounts of sacrifice and cannibalism throughout South America have not been confirmed by any archaeological record. Quotey voice again. Rather than an example of violent death and careless deposition, Playa Venado presents an example of how pre-Columbian societies showed respect and cared for the kin after death. So this article is also a Smithsonian Institute co-author archaeologist Richard Cook. It was published in Latin American Antiquity. So the 1954 paper also had a catchy title. Lothrop's paper was titled "Suicide, Sacrifice, and Mutilations in Burials at Bernardo Beach, Panama." It's been cited 35 times as evidence of violence, cannibalism, trophy decapitation. Some authors have used the paper to suggest that it is a mass burial site or manifestation of conflicts at the time. Now, this guy in the this guy's studying this in the 50s, which is actually before bioarchaeology, the study of remains from archaeological context, was even existed. That showed up in like the 80s and 90s when that became an actual field where you would determine what took place. And it has been updated even since. Like, this is an ongoing thing. And, he found a bunch of bodies. The bones looked broken. They were broken. They were broken in many places. The heads were sometimes separated from the rest of the body. It just turns out that happens naturally to buried bodies. Also, there was evidence that these bodies had been uninterred before, that they may have been moved around. So you bury the family in the family plot, but then the family moves. You take your dead with you. You're not going to leave your dead behind.
Yikes. That makes moving day more complicated.
They make it a lot more complicated. They also found that some of the graves had been disturbed to add other bodies to those graves. So, one matriarch passes, the patriarch is added to the grave later. It's that sort of thing taking place as well. Broken bones actual broken bones in life were present in many of the bones, but they had healed, meaning that they weren't the cause of the passing. So, yeah.
I think it's perspective, and it's just another example of our changing ability to have a different perspective based on what we understand, our improving resolution, our ability to see things. It's just looking at things and going, oh, let's not tell this story of, hey, we need to have conquest. Let's tell the story of the actual people. Hey, look at those cracks in the bones. Oh, they were broken, but healed before death. These aren't death trauma things.
It also means that they were cared for.
They were cared
by their
for.
community.
They healed.
When injured, because they were allowed to heal. So yes, there were very brutal people in the past in South and Central America. But chances are they had Spanish last names.
This very much reminds me of how I was just talking to someone recently about scalping and Native American certain tribes that were accredited with starting that tradition. And there's now new stories coming to light that suggest that potentially white people did it first, and then the Native American tribes took it from there.
And said, hey, if you're going to do that to us, we'll do it.
How do you like it?
How do you like it? Yeah.
So it's another situation where we want to kind of separate ourselves, but really it turns out maybe we're more similar than we realize. Maybe they're not so different.
Yeah. And sometimes it takes a long time for the evidence from cold cases to tell the story, the true story.
Absolutely.
Really long time. And gosh, we got that horrible, awful email about all of the things that were lost to the linguistics department in the Brazilian museum fire.
Yes. Yeah.
They had, these are languages that no one speaks today, that they had recordings of that burned. So not only is not only were people made voiceless by conquest, but even those sounds of their voices, those dialects and those languages themselves are now lost to us.
Yeah.
Really horrible.
Yep. Oh, such a.
We need a better. We need a pick me up segment. Do we have anything?
I
What
don't know.
No.
Do we have
No.
anything
That
like
usually
that on
ex-
this show that kind of
less of
picks
a
us
downer.
up, makes us
Are
feel
you
a little
implicating that
happier?
I am the fluff piece in this show? Because I do not appreciate that.
I don't know, but I know it's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
Got Blair.
I do have a theme song. It's true.
You have a happy theme song, and it's the animal segment.
I mean, touche. Well,
Let's
my
go,
first
Blaze.
my first story is about studies done with animals, related to animals, including animals like ourselves. Did you know that when you're sick, you smell different? And it's not just because you were too tired to take a shower. There's actually a smell of sickness. This is known in the animal kingdom. It helps animals separate from animals that might get them sick, so they themselves do not get sick. This has been previously studied, but a new piece of research from the Monell Center looks at the significance of personal odors as a source of information about individuals' health that are healthy, but hanging out with sick animals. So this study found that bodily odors of otherwise healthy animals sharing an environment with sick animals became like the odors of sick animals. So even if you yourself are not sick, if your roommate or your significant other or your child is sick, you might carry the stink of sickness.
Wait a second. But I'm not sick.
Yes.
How did I get the stink?
Great question. Exposure to the odors of sick individuals may trigger protective or preparative responses in their social partners to minimize risk of impending infection. So this odor has actually been found in previous studies from sick animals to stem from inflammation. Inflammation leads to these odor changes, which suggests that immune-activated odors might signal the presence of disease risk in the sick animals. Here we consider that perhaps being near sickness causes inflammation and therefore the smell, even if you yourself are not sick because you're fighting it. You're like bulking up for a potential fight with an illness.
This is brilliant. So I am going back to studies that we've talked about in plants, where plants communicate with each other through the air. You know, when they are bitten, where they're being eaten by other animals, there are compounds that get released to let other animals know to release more bitter compounds.
Yeah,
This
right?
is such a good connection. It
There's
didn't
a
even
biter
occur
in the
to me.
neighborhood.
Yes.
This is the chemical signaling of damage.
Yes.
That is
absolutely.
And so this is going into other kingdoms, right? That this is an old signaling pathway.
Wow.
Chemical signaling to prepare, or it's this chemical communication, basically.
Yeah.
Sender receiver.
So here,
there's
yes.
there's another element to this too.
Okay, let's see if my further research answers your question. I'm very curious.
Because what do predators tend to try to pick out of the herd?
The sick and the weak.
Right. Now, I also smell like I'm sick. But I'm in the prime of health. Why don't you chase me instead? I smell sick. Come and get me. But I'm going to get away because I'm fully fit right now. But now I've sent out a signal that confuses the predator's ability to pick which one out of the herd.
I think if you're a prey animal, you don't want to be chased.
You don't care about your family?
No, you don't.
Even if I don't agree.
I wholeheartedly think that even if you're a healthy zebra, let's say, you do not want to be chased.
But your genes do. Your genes might differ. Your genes don't care what you want. They're there for the gene pool.
Fair enough. Well, I don't know. But this
That's
research
self-esteem theory. That's good. Yeah.
So this research, they had a few different elements of it. So this was done in a lab with mice. They injected mice with a non-infectious bacterial toxin that causes inflammation. So this in theory is not contagious, right? So they make quote unquote sick mice that then create this sickness odor. Then the injected quote unquote sick animals were housed in cages with healthy animals. They found that these mice, that sniffer mice, who then were sniffing assays from these animals, could not differentiate between the sick and the healthy housed with the sick. Then they repeated the same experiment where the sick and the healthy animals were physically separated by a perforated partition. So that allows odors to circulate, but no physical contact. So that means there's no physical odor transfer because they're like,
they're not rubbing on each other.
Yeah.
It's not a grooming thing. It's not, yeah.
No, so just
It's
the
through the air.
sickness odor created this inflammation and sickness odor in these other mice. So a couple things here. This can affect a lot of population dynamics, kind of what Justin was talking about. If you're thinking about predator-prey reactions, if you're thinking about social interactions within a species, this can affect a lot of things. But it also informs efforts to use bodily odors to understand how pathogens are transmitted. So if you know you're avoiding animals that have been around a contagion, then how do these diseases spread? So they kind of have to spread quickly, right, before these pathways, these odors show up. So it's pretty interesting.
But I don't think we're maybe consciously attuned to it. Or even like humans, I don't think might be.
Not consciously, but you definitely know. Have you ever walked into a house where people are sick and you're like, oh, there's something off. We are visual animals, but we do have a very good sense of smell still. And we are, if not, as you say, consciously attuned, unconsciously, our brain is responding to signals.
Yeah. But
we can exactly. We can top down override those signals if we choose to, and we do all the time. It's like, I'm going to work. Even though I know Bob is sick, I'm still going to work. Even though
the person who handed me a coffee just sneezed. I want my coffee more than I can get a coffee.
Yeah.
So I can see like it being really hard to translate this into the human animal world in terms of how we interact. But yeah, like if you're part of a brood and one of the birds is sick and it's getting transmitted, that could affect breeding if it happens at the wrong time of year. It could affect your whole family, your whole group has now been like, we're just going to stay away from all of them until that either works out or doesn't. So that is a form of an immune system within animal behavior.
So someone in the chat room brings up a really interesting point too. If you work in the emergency room, do you always smell like a sick person? That's interesting.
But the question here also, I mean, this study, it's mice and it's also looking at the urine of the mice. It's not like capturing the odor of the ode animal, whatever senses are coming out of their skin.
Everywhere.
Yeah, which mice do.
Mice do. They mark everything. They urinate all over. So and in humans that could potentially, it's going to be different. The human animal is definitely different.
Because, I know when I've got a cold.
But the question is,
pee changes smell. Like I can tell. Like I'm like, oh, that's a cold pee. I'm coming down with it for sure. I recognize that. So yeah, that would make sense. But it's hidden not because of the subconscious or the conscious mind. It's because we don't pee everywhere.
Well, I would also argue that as animals with facial features, no fur on our face, all this kind of stuff,
body odor,
we carry other signals too.
Yeah.
And so even if we might have this odor-based system, we now have this whole other laundry list of signals that we're giving out. I mean, we've all been, we've all walked into work and someone's gone, "Oh, are you sick?" And you're like, "No, Karen, I'm not sick. How dare you? I'm gonna go look at myself in the I'm not sick." But like, how rude.
You're looking very tired today.
Yeah, you look tired. It's like, first of all, shush. But second of all, for some reason people, have the verbal vomit and they say these very rude things, but it's because you're carrying this on your face. And there's biological reasons to tell people if you're tired or you're sick.
Yeah. And like I mentioned, we have body odor where mice don't have, armpits in the way that humans do. They don't have the same kind of body odor that we've got. I mean, and you can eat things that change your body odor. When I eat curry, I think I smell like curry. It sticks around for a while. But that odor, if you are ill, especially if you're very ill, your body odor is going to be different. So
Absolutely.
yeah, I think there's a lot more to be looked at in this and to these unconscious messages that affect our behavior.
There you go.
Chemical communication between animals. I love it.
And now I have two very quick stories that are very related about animals with expected diets and these particular animals deciding, no, I'm going to eat whatever I want. So just like we've talked about the show with deer and other supposed herbivores that have been found to be just forever eating meat, and we just haven't known where to look. But hey, that makes sense because these deer live in a space where it's frost and ice and snow for months at a time. How could they possibly be living off of grass? There's no grass. So it turns out, they're scavengers.
Wait, what? This is
the
first
first
time
time
I've heard that.
No, we talked about the show years ago. It's just been so long. It was probably like 200 episodes ago.
That's why I have an old brain.
Yeah.
Deer will eat like an old squirrel that froze to death?
Yes,
absolutely.
Yes,
yes, they will. Yes, I remember this. I remember this story very well.
Just as supposed herbivores might occasionally eat some meat, sharks, the carnivores. If you think about a meat-eating animal, a hunter, a savage, what do you think of? You think of sharks, which, is a whole nother issue. But it turns out that bonnethead sharks actually eat seagrass. Yeah. So this study looked at, apparently we knew that sometimes seagrass showed up in the gut of bonnethead sharks. We weren't sure if they were digesting the stuff or if it was just kind of like they were thrashing around. They ended up with grass in their stomach, which seems unlikely when you look at the gut content, which is about up to 62% seagrass. Now it's looking more intentional, right? They found in this study that they actually can digest it. They are assimilating seagrass carbon, and that the cellulose component degrading enzyme activities were detected in shark hindguts, showing that this coastal shark, the bonnethead shark, is digesting seagrass with at least moderate efficiency. And that means that our ecological expectation of the role of these sharks is completely different than we thought. They are omnivores. They eat meat and plants. And so their nutrient transport in the ecosystem is completely different than we thought it was. So sharks can eat plants and praying mantises.
Well, I was going to say one thing with the sharks eating the grass. I mean, the important thing here is it's not like the, pandas where these are carnivores and they don't have the right enzymes to digest the bamboo.
Oh, yes, they do.
Right.
Yes,
They do. They do. But
they don't have the length of an
intestine,
yeah.
Right? But these sharks do have the length of an intestinal tract. The digestive tract is long enough, and the big thing is they're not just eating grass and it's sitting in there. They have the enzymes that are capable of digesting cellulose.
Yep.
Which we all know is that's the big thing. We got to, all sorts of animals have figured out ways to digest cellulose. Ungulates have fermentation. Other animals have enzymes. Right. Which is why fiber is great for your digestion.
Yeah, yeah. Just scrubs it all those fingers. Scrubbing all those fingers we talked about before.
Rubbing all the fingers.
Yeah, so they're not just
really
eating
fast.
it, they're actually digesting it. So these sharks, true omnivores. Praying mantises, they, forever we've known, are mainly predators of insects. There have been occasional anecdotal accounts of them feeding on vertebrates, including small birds, lizards, frogs, newts, mice, snakes, and turtles. But they have always been one-off accounts, one instance, one animal in a lab or some sort of human-manipulated environment. So the idea of whether praying mantises are feeding off of vertebrates in the wild, we have no idea. It's never been observed. Until now.
Huh?
A new piece of research published this week in Journal of Orthoptera Research found an adult male praying mantis hunting and devouring guppies in a pond in a private roof garden in India. So this is in the wild.
Very
it is, I will
specific
agree,
type of wild.
a
a roof
human
garden
manipulated
wild.
environment.
Yeah.
Look at him go. Look at that savage. Curiously enough, the predator came back 5 days in a row to catch 9 fish, at least 2 a day. This insect would walk on the leaves of water lilies and water cabbage on the surface of the pond to then grab the fish. So this is a repeat offender in the wild, eating a vertebrate. So this is a first ever. And this opens a bunch of new questions about praying mantises. So one, this confirms that they can indeed eat vertebrates and that therefore their impact, praying mantis impact on an ecosystem, is very different than we thought. We thought it was just bugs. That's it. Now, if they're attacking and eating vertebrates, that completely changes the story of their role in an ecosystem. Yeah. The second, this is really interesting. We had always thought that praying mantis's eyes, those amazing eyes, were developed. They have adaptations for hunting in daylight. They really didn't think that these eyes were well equipped for hunting in the dark. But this specimen, each time, caught fish at sunset or late at night in the dark.
Interesting.
Yes. So our expectation about the visual acuity of mantises, totally different.
Yeah.
And
so that would almost indicate the opposite of what we thought, that those eyes are actually a little bit more effective when the sun goes down a bit and it's allowed to the bright light to see the contrast and the differentiation and track the fish and go get it.
Yes.
Yep.
And the third and perhaps the craziest thing that we've learned from this account that we've seen that now requires further research. It doesn't identify anything as a truth because it was one mantis, right? But this male mantis learned some sort of learning to know I can step on these lily pads to get to this space. There's food under the water that I need to catch in the only way you can catch a fish. It's not the same as catching bugs, right? So this is some sort of cognition that helps them to develop novel hunting strategies. So either this is from personal experience, or it learned it from another mantis, or it stumbled across it. We don't know, but for some reason,
exceptional mantis that we've ever
observed,
which is always possible.
I think the third thing that we're really learning here, though, is that our fear that praying mantises may take over the world is warranted. They are the aliens we should be fearing.
That's right.
I'm afraid
That's right.
I may be rooting for it.
Yeah, but yeah, these guys. This one dude came back 5 nights. He kept coming back for those delicious guppies. So something about it, he learned. He came back. So two animals completely breaking the paradigm on their diet and their food acquisition methods.
Yeah, and especially with the mantises. I mean, this is probably going to provoke a bunch of research into the visual system beyond what already has been done.
To the
Yeah,
visual system.
That's
and
interesting.
yeah, because if you've
Yeah.
been studying this bug, maybe you clock out right before sunset because nothing to see here.
Nothing to say here
Yeah.
except for
They don't do
fishing.
anything. I learned that in school. They don't learn anything. They don't do anything from here on out. So let's just go home. But the other thing too is
Oh,
like it makes me think like, what if this is the first fisher mantis? Right. This is the cusp of new knowledge and new technology and new adaptation and a new and then maybe it's happened 100 times, 1,000 times before, but that it's hard to teach the next generation to do it. So they're all one-offs, these
Yeah, they're
discoveries,
one-offs.
right?
Yeah.
Interesting.
Yeah.
well, we will leave you with these thoughts for just a few moments, everyone, because it is time for This Week in Science to take a break. We will be back in just a few moments after some messages with more science. We've got some other really great science lined up for you in the second half of the show. Please don't miss it. Stay tuned for more.
Can't explain things you've heard with more than intuition. A lot of research shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within.
Scatterplata. Hey everyone, thank you so much for joining us for another episode of This Week in Science. We are back again thanks to you. Yes, the fact that you listen to this podcast, the fact that you watch us on YouTube, the fact that you interact with us, all these things. It's your support that keeps us going. So thank you for that. We are so glad you enjoy this show. Now, for those of you who would like to know how you may go about supporting This Week in Science, what can you do? How can you help us even more? What can you do? Well, the first place to find answers to those questions is at twis.org. Please head to twis.org for information about This Week in Science, for latest episodes, and to subscribe if you have not yet subscribed, or if you know somebody who really needs to listen to us on a regular basis, tell them to head to twis.org. They can click on the subscribe button. It's a big orange button. You can't miss it. And that will allow them to choose their proverbial poison: Google Play, iTunes, YouTube. All three subscriptions are wonderful. They let us know you're out here, right? That you're out there listening. Be one of the numbers. And then while you're at the page, you can also peruse our store, because there is a place in the world where you can find lots of goodies that have the TWIS logo, that also are emblazoned with art from previous Blair's Animal Corner calendars. Lots of wonderful things. Do you need a cup for your coffee? A mug? Well, there is a basic logo mug in our store with the TWIS logo on it. Do you like t-shirts, but hey, a polo shirt would be better for your needs? We have a TWIS logo polo shirt. We also have phone covers. We have pillows. We have mouse pads. There are hats to guard your eyes from the sharp, bright glare of the midday sun. There's also wrapping paper for those upcoming holidays. So many good goodies. That's right. Does somebody you know love TWIS? Maybe there's some holiday presents in here. Buy someone you love something related to TWIS and support TWIS at the same time. A portion of all these sales go back to us to keep everything running. How else can you help keep everything running? Well, direct donations are that way. You can click on the yellow donate button at twis.org. It will take you to a PayPal interface where you can donate one time and any amount you like, and it's super easy. Also, if you go to any episode page and scroll through the show notes, lots of neat show notes, information, links from the show you can find there. If you ever want to know more about a story, that's where you can go to find it. But at the bottom of the page, there are pink buttons that can allow you to donate in an ongoing fashion through PayPal. That's right. Set it and forget it. $2 a month. It's a really easy donation amount every month through PayPal. So you don't have to think about it and remember it. It'll just happen. $5 a month, $10 a month, whatever you are able to do. It is appreciated. If you don't like PayPal, however, you can help us and join us on Patreon. Patreon is a platform kind of like Kickstarter, but it's more like for patrons of the arts. And you can choose whatever level of support fits your monthly finances. And you are charged once a month in a recurring fashion once you sign up, uses your credit card, I believe. And all you have to do, click the patron, become a patron button. It's a big red button. Click it, become a patron. How about that $10 a month tier? The $10 a month tier, we will thank you by name at the end of the show, in addition to other wonderful gifts. $10 a month. Pretty easy amount of money there. You spend more on coffee during the month for sure. And I hope we give you a little bit more than that coffee does in terms of satisfaction, sustenance, and knowledge. There we go. All right. This week in science, those are the ways that you can help us. TWIS.org is where you can find links to all these wonderful things. All of you out there, you are the people who help us keep this show going. And really, if it weren't for you, we couldn't do any of it. So we thank you for your support.
Can't explain things you've heard more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need.
And we're back with more This Week in Science, everybody.
Yay, we are back, and it is time for our segment of the show that we love to call This Weekend. What has science done for me lately? What has it done? Oh, I have a letter from Todd Barnell. He says, I know you and Blair had some concerns about the whole vaping thing a listener mentioned lately on the recent What Has Science Done for Me Lately, but I got turned on to your podcast about 6 months ago by a friend, and now TWIS, Science for the People, and Story Collider are highlights of my week. And for 35 years, I've loathed my addiction to nicotine. Oh, if I could go back and smack the 15-year-old me upside the head. I've spent the last 2 years especially trying everything out here with no luck. And then I heard the latest podcast. There was no way I was going to join the annoying hipsters with their idiotic vaping sticks until I listened to you read that guy's comments. So I went to my tobacconist and all red-faced told him I was thinking of trying vaping as a way to quit. He grinned and said, dude, that's how I quit a year ago.
Wait, you're tobacconist? Your tobacconist quit?
That's right.
If that guy can do it, anybody can.
Yikes.
He's a tobacconist.
That is how I, let's see. And now I have massively reduced my nicotine intake. I can feel my body slowly weaning itself off in a way I can handle. I can see the light at the end of the tunnel. And heck, even if it takes a while, I am vaping, not sucking on non-filtered cigarillos. That has to be a win. And in a week, I saved $20. So I just sent that to you all. And the store manager added TWIS to his podcast list, by the way.
Fun.
Awesome. Thanks for an amazing podcast. Thanks for filling my brain with amazing, beautiful, inspiring things every week. And thanks for turning me on to a tool that just may help me in other ways. Cheers.
Fantastic.
Cheers to you, Todd. I think it is. I'm glad that science and what has science done for you lately has helped you.
Yeah, that is pretty cool.
And so everyone out there, your notes, the things that you write about, other people are hearing them and they may have downstream effects that can help other people. So if you have wisdom, if you have beautiful thoughts, if you have whatever thoughts, share them with us. Maybe, well, who knows what may come from your story of what science has done for you lately. Send me a message on our Facebook page, facebook.com/thisweekinscience, or email me at Kirsten, KIRSTEN, at thisweekinscience.com. Help us keep filling this segment of the show with your stories. Please help us do it. All right, are we going to keep going with more science stories?
No, let's call it a yes, of course.
Show's over.
I was talking about.
I'm going home.
And I'm spent.
You're already home.
That's it. All right, Justin, what do you have for us now?
Oh, it's my turn. You know what? You know, it is kind of getting late. Maybe we should. Oh, wait, no. Here we go. Scientists have known for decades that certain rod-shaped bacteria produce small spherical versions of themselves. These mini-me microbes that they create actually don't reproduce. They don't seem to do anything. They're like, it almost looks like you started to reproduce and then called it quits, like partway through. And so you get this little spheroid instead of the regular rod-shaped bacteria. So there has been a lot of interest in these little mini-me microbes because they can actually be infused with drugs and then put back into tissues, and they go around, they interact with the tissues, and they can deliver those drugs. So there's been some study going on, but the actual role, like why does this happen, has been a mystery. Nobody knows why it happens. And that's the story. No, wait, there's more. Researchers at the University of California, San Diego have discovered that the mini cells actually play a pretty important role in self-preservation of the bacteria. Publishing their results in the journal M-Sphere, researchers discovered that E. coli bacteria discharge damaged proteins. They bundle them up inside these mini cells, and when the mini cell is full of these damaged proteins, they toss them out. This is, I'll do quotey voice. This is Camilla Rang, research specialist in Division of Biological Sciences at the laboratory. It's amazing that even bacteria take out their garbage. We have shown that mini cells can be beneficial for the bacteria and help them escape death by kicking out the damaged proteins. Also, what's sort of interesting about this, a lot of antibiotics, the way that they're mechanically, physically operating on a bacteria, is they're targeting proteins within that bacteria and damaging them. So if the bacteria can take that damaged protein and toss it out, that might be a path to surviving an antibiotic. So to examine the links between mini cells and the health of bacterial cells, researchers tagged and tracked proteins with fluorescent markers. While under attack from antibiotics, researchers used microscopy techniques and, excuse me, to follow the damaged proteins, and they saw them package them up, kick them out. Sort of like one of those sci-fi movies where you got a spaceship and it's like, it's under attack, and then the little escape pods launch out. That's kind of what it looks like. Out they go.
Garbage. It's the trash escape pods. I'm out of here. Antibiotic away.
So the other aspect of this is that they may then find another path to putting therapeutic drugs in targeted places, because the method currently is trying to infuse these little mini cells with the drug and then injecting them back into the body, because they can still interact. Even though they don't reproduce, they can still interact with the body and deliver those drugs in targeted ways. But now if you could get the bacteria itself to produce your drug, and package it up and put it into the little. And now you've got a system that can replicate. So a lot more research to be done. Kind of a nice note at the end of this. They dedicated their paper to William Loomis, who was a 50-year UC San Diego faculty member who provided critical input to the direction of the research project, but passed away in 2016. So they remembered to mention the person who, although didn't survive to be part of this paper. It's a nice tribute.
That is a nice tribute. Absolutely. Remembering the people who were involved and gave probably some really important insight. And this is fascinating. I love that these microbes are able to, package things up and get rid of them. But I'm wondering, last week talking about what was it, the antidepressant that it possibly affects antimicrobial, antibacterial resistance. And part of it affects the efflux of the antibiotics, helping the bacteria pump antibiotics out of their system. So I wonder if that would be something, help them package it up into trash as well.
Yeah, it's
similar. I
also
wonder what
interesting.
else
Like
is involved in that.
This is something that's becoming this budding system that is like the only real system that these microbes use to send stuff out or to reproduce. Even it's also conserved in neurons, as we've talked about. But the concept of garbage removal. We've also talked about this in terms of the lymphatic system that surrounds the brain recently, like
right,
trash
Alzheimer's disease.
Yeah.
It's stuff getting stuck and not getting removed,
Not
right?
getting- So this trash removal within that thing that we humans do when we take the plastic bag full of garbage and put it in the container and then push that out to the curb once a week. That's not new. Life has been doing exactly that action from the conserved from the very beginning. We're really not that different.
Everyone's got to have a good garbage disposal system. That's right. Get rid of the garbage. We have to figure a lot of that out as humans still. But anyway, there's other litter in the world besides trash. This litter is in our DNA.
Huh?
Yeah, our genome has the refuse, the detritus, the remains of ancient retroviruses that inserted themselves into our genome. Maybe they got copied. Maybe there's all sorts of these retroviruses that exist within our DNA. And some of them have, we know, gotten involved in the expression of proteins, the expression of genes. We know, for example, our mitochondria are, these, they used to be bacteria, but they became endosymbionts. They became part of our system. And there are many mitochondrial based diseases because of genetic mutations within the bacterial DNA in the mitochondria. So there's a lot of the potential of disease from these ancient alignments within our genome. And there's a new study that just came out where a team at Oxford University and the University of Athens looked at a retrovirus that is in humans. This invaded the germline of our primate ancestors a long time ago. And it's called HERV-K-HML2, otherwise known as HK2. Thing is, many times these retroviruses end up in our genomes and end up conserved, and humans all have the same sequences for these retroviruses. Like they're all the same. However, in HK2, there are a lot of variants within the human population. And so the researchers for this study wanted to know how the variants of HK2 might be associated with human disorders, human disease. And so they analyzed samples of hepatitis C virus infected patients from the UK and HIV infected patients from Greece who had become infected due to known infection routes, so injecting drugs basically to determine the frequency of this HK2 integration into individuals. The HK2 insertion, where it exists in this particular gene in our genome, is usually prevalent in only 5 to 10% of the population. However, they found that within this population that they looked at, these samples that they looked at, people with well-defined addictive behavior, like injecting drugs, are 2 to 3 times more likely to have this HK2 gene within integrated into their genome.
So wait a minute. Does this mean that there's something in the community of drug users that gets this gene to insert itself, or does this mean that this retrovirus likes drugs?
No, the retrovirus doesn't like drugs, but it is inserted within a gene called RAS-GRF2, which is involved in regulating dopamine activity, which is part of the quote unquote pleasure seeking pathway, which is associated with addictive behaviors when it is kind of overstimulated or it responds abnormally. And so what they are hypothesizing for this endogenous retrovirus is that there are certain variants of the retrovirus that can affect human behavior. That HK2, some variants of it, because they're and the variant and the location within the dopamine affecting gene may be influencing addiction. And so addicts, you can go backwards, addicts may become addicts or become drug seekers because of a retrovirus in their genome.
Wild.
Isn't that crazy?
Yikes.
An ancient virus stuck itself in the genome in just the wrong place, and hello, you inject drugs.
But here's the other study that I think would be interesting to do in here, because I don't know what the demographics look like for this, but those retroviruses that we all share. Oh well, some ancient common ancestor to us all encountered this and had it inserted. Fine. But for only a small percentage of the population to have this virus means that there was an event that wasn't a universal, wasn't the ancientest of ancestors that, could have led to all of us, but that it's in a pretty small, if you think about it, subset of the population that encountered this. We should be able to track all of these people back to a common ancestor at a place in time.
Yeah.
And fascinatingly, because this is the 5 to 10% population, that's going to be a rather specific backtracking because of all of the genes you would have to choose between this gene and that gene every time. But still, that would be fun anyway.
Yeah. So what they are saying is that this is for the first time strong proof that these human endogenous retroviruses can be pathogenic in that they can cause disease or disorders. For the first time, we are able to make a distinction between cause and effect in HERV, human endogenous retrovirus pathogenicity. And then they also say looking into this dark part of the genome, which is basically this not well understood part of the genome, these retroviruses, will unlock more genomic secrets.
And it only sounds like it's become pathogenic in the face of hard narcotics. Like, we don't know. There's some level of possibility that this was a beneficial thing to have if you at some point in this history, which would also be part of the story. It's just in the current modern age that it's pathogenic.
It
This
doesn't,
could
yeah,
have
it
been
doesn't
a
work well.
fantastic benefit at some point that allowed that small population of humans that grew to 5 to 10% of all of us to succeed. We don't really know.
Yeah, from the Oxford press release, they say most HERVs in the genome are shared by most human individuals. So most of them are beneficial, or at least are not disadvantageous. They're not disadvantageous, and they spread through just about everybody. And so if you pick 2 random people and check to see if they have the same HERVs in the same genomic location, it will be there. This is not true of some copies of HK2. There's variation, with certain individuals carrying extra copies in their genomes at distinct locations. Amazingly, it appears there are some rare variants where the integrated virus is coding competent. In other words, it is an unbroken blueprint that could be used to make more viruses, which could be active and cause problems to the human host. So this HK2 is fascinating.
Wait a second. So this means
unbroken, possibly, where quote unquote coding competent could be.
We'll put that, but that's even a possibility that
So not only is it just interrupting and affecting this the dopamine pathway through gene transcription and translation, it's this gene in some people, this retrovirus in some people, if it's not turned off by genetic instructions, it could be releasing virus into some people still.
Jeez.
With who knows what effects.
Right. That's very wild.
Yeah.
But I also suspect that it takes place more than we might think. I always wonder how it is that infants end up with the right viral load to do the bacterial screening if they're not producing it through their DNA. Like, that has to be how that happens, right? Or maybe wrong.
Maybe not. Nope.
Yeah, the other possibility.
All right. Do you have another story?
Yes. In new news about what goes on in a nose, recent research by researchers has revealed that the mix of bacteria that lives inside of your nose correlates with the type and severity of cold symptoms you will develop. Yes, it's a correlative find. This is where science often starts. But people whose noses are saturated with Staphylococcus had more severe nasal symptoms than cold sufferers who had less staph. Okay. Well, you say, yeah, but you got a staph infection. No. They have a cold that is being created by a virus strain. They have the same virus strain in each individual. But there's a correlation between what the microbiome of the nose is and how that cold affects them. So they actually were able to find that the bacteria in volunteer noses fell into 6 different patterns that they refer to as buckets of nasal microbiomes. And the different patterns were associated with specific differences in symptom severity. But what comes first? The nasal microbiome or the cold? Or is that how it is? It because is there an environmental thing? Is it an affect of well, they decided to take a look at it. So this described they are described as having been very surprised by this finding. The first surprise, and then they go on to tell you that they were surprised. The first surprise, this is Quotey voice if you can't tell that I shifted. The first surprise was that you can kind of identify these different buckets that people fit into. And then the fact that the buckets seem to have some impact on how you respond to the virus and how sick you get was also interesting. This is Ronald B. Turner, MD, University, Virginia School of Medicine. There were effects on virus load and how much virus you shed in your nasal secretions. So the background microbiome, the background bacterial pattern in your nose had influences on the way that you reacted to the virus and how sick you got. Well, sort of, because then they're going to take that right away. So the nasal neighborhood of microorganisms are not the cause of any of these colds. The colds themselves were caused by a cold virus that the volunteers voluntarily took after their nasal microbiome was checked.
Oh,
So,
yikes. I would not sign up to get a cold.
right, you kind of have to be in the right place in your month, week, life. Actually, I got the
A
next
college
couple weeks
student
off.
real hard up for some beer money, maybe.
I don't know how you really choose it, but if somebody says it's for science, I think I'd do it. So anyway, so
maybe it's because your colds aren't as severe as mine are, and so you wouldn't mind as much. So your nose bacteria is different than mine.
Yes. But does that mean that we would be affected by a cold differently, according to this? Yes. So what we're reporting is an association. So it's entirely possible that the fact that you have staph in your nose and they have more symptoms is not directly related. It may well be that there's some underlying host characteristic that makes you likely to have staph in your nose that also makes you more likely to become ill. So the correlation, they didn't have it drilled down. So, what do they do next? They give an antibiotic. Yes. So they had 152 participants in all. So this isn't even a small group of people who raised their hands.
That's a lot of people.
It's 150 people who were like, I don't need to do anything this week. So they tested the nasal microbiomes before giving them the cold. They give them the cold, and then they gave people probiotics. So they found that after getting the probiotics, the changes that they saw were none. Nothing.
Probiotics or antibiotics? They were wiping them clean, or they were adding bacteria?
Great question. And if it was a terrible question, I would have told you it was a terrible question too, just to be fair. But that was a great question because that's what they proposed doing next. The probiotics didn't have any effect, really, even on the gut microbe because they didn't wipe it. It didn't change the nasal microbiome at all. Didn't change symptoms, didn't change outcomes, no difference. But they do postulate that they need to redo this study where they do use antibiotics. It's just tougher to do because they don't, even scientists doing research don't like to give people antibiotics for colds because of the day and age we live in. But they're saying that's the next step, to start with the antibiotics, do the cold, and see if that same
if it changes it.
Yeah, see if that changes, and that you get a different result from there. And then, so
I'm not surprised. I'm just going to say I'm not surprised about the probiotics not having an effect. However, because there have been lots of studies recently that have kind of suggested that probiotics don't work.
Well, okay, so
sorry, yogurt.
So there was so the difficult thing about talking about probiotics is you're talking about just adding them to a very crowded system.
Yeah,
where niches are well defined, where roles have been in play, possibly for decades. They're not going to sweep the if you've been given antibiotics and you've had that system
changed and then
out.
Right.
Yeah, then you're replenishing. And what was interesting, and I didn't bring this, I won't talk at too much length. There was a study I read recently that the order in which you add, if you add one at a time, these probiotics to a clean system, will determine what that final microflora will be. So if you change the order that you introduce them in, for instance, you will end up with a different stable system at the end. So even though probiotics and the microbiota is the cutting edge of knowledge in science, it's the cutting edge because we don't know what's across the cut. Right? We're still learning a lot.
So I have an anecdotal account that I think is related to this. Maybe, who knows? Which is that I used to get the entire time growing up, I used to get, when I would get colds, the worst sinus symptoms. I would have a runny nose, blowing my nose, couldn't breathe through my nose for weeks.
Wow.
And I had this very hippy dippy yoga instructor in college who I came to class once with one of these runny noses. So this persisted into adulthood. I was a young adult, but I was still definitely full grown at this point. And she suggested I use a neti pot.
Right.
Now, we've talked
Right.
about this on the
Yeah.
show before, the neti pot and the pros and cons and all this kind of stuff.
Mhm.
I use that thing religiously twice a day for a month. And I have never, knock on all the wood, had the same responses I used to have to colds that I had. Now, I could have just grown older. There could have been other things that happened. It could be totally anecdotal, but it's interesting that potentially could have changed the balance of things going on in that space.
And you would
Absolutely.
be a perfect test subject then to be given a cold virus and to maybe add one of these microbiomes to
Yeah. Yeah.
see if that could reverse what the neti in your
Reverse it.
yes,
No!
you are the perfect test subject. You have to now go and volunteer and get this cold because
No.
I guess the question is at this point, I mean, scientists, can we recreate what has been created in nature, right? Can we figure that out?
So maybe in the next stage of this, when they give antibiotics, they also try a sinus rinse version where they're physically trying to remove or change the
And that wouldn't
community
be
that's in there.
antibiotic, but
Yeah.
it would be a physical, neti pot.
Would change
We
it.
should talk to them and tell them what they should do for their next study.
Yeah. Justin, get him on the phone. Just dial on your old rotary phone right there.
Well, there was one
Then-
thing that I did think was sort of interesting is that they
Hello.
the probiotic was a drink.
There's no reason that should help your nose. Oh my
yeah,
goodness.
like so the
Like
idea would
stick it
be
up
like
your
a nasal
nose, guys.
That's what you got to do. That's where it's at. And the researcher was like, yeah, we considered that, but we didn't think it would be effective. Try it. That's how you know.
So
Here's
there's
some yogurt in your nose.
so they do
I
have
know.
nasal
Okay, kids at home,
spray,
if you're
right?
listening,
There's
do
like
not put
instead of a
yogurt in
flu
your
shot, there's a flu nasal spray, right?
Mhm.
So you could get a probiotic nasal spray. That's totally possible.
For sure.
And in fact, you
sure.
can get them at Dr. Justin's, not a real doctor. Nasal spray. It'll
Now
put
you-
bacteria where your nose is.
Is it yogurt in a
Mother-
hairspray can?
Don't give out the secrets. Did you sign the non-disclosure? Or was it I think it was implied by us talking.
Moving on. Here's a study out of the journal Science from this last week. Researchers inventing new biology. That's right. Oh, synthetic biology. We're trying to create synthetic organisms. One part of that is we basically need synthetic control systems for these organisms. Let's put together entire systems of enzymes and the controllers that work with them. And so in the laboratory of Michael Elowitz, he's a professor of biology and biological engineering and a Howard Hughes Medical Institute investigator, they have been figuring out how they can create a synthetically engineered toolkit, kind of like Legos for the cell, to program new behaviors for cells. Yeah. And so normally to change behaviors, we edit the cell's genome, right? There's gene editing, and that can get passed down
Because-
from generation to generation.
Because handing a hand tool to each microbe is a painstaking task. You want it to make its own. That's why it has to be in the genome, right? Like it has to already
but
be there and
sometimes-
have it be making it.
Yeah, but sometimes you don't necessarily want something to just spread forever through the population, right? You want to have the ability to turn it on and turn it off. So you want to have molecular control switches And so, according to Elowitz, a major goal in synthetic biology is to use less genetically permanent solutions. So it's kind of like, semi-adhesive tape and not super glue, according to their analogy.
That's a good one. I like the idea of handing a kid drumsticks and then being able to take them away again.
Exactly. Yeah. So they're creating, they're calling them kind of like circuits, but they're not circuits. They're series of molecular instructions that can carry out a function and then go away once they're not needed anymore. And so the researchers in this lab at Caltech, have developed a set of proteins that can actually be used together, kind of like building blocks wired together to sense the environment and act according to the environment, and then be taken apart and not work when they're not needed anymore. And so ultimately they want to create systems that diagnose and respond to specific cellular circumstances.
Wait a second. So what they're doing is they've created their own enzyme, and then they create their own protease. They just add that later.
Exactly. Yeah, they're creating.
Cool that
it's like cells already have these things, but if you want a cell to do something new that it's never done before, you're creating that behavior. You're creating the tools for the cell to do the thing you want it to do. Yeah. You have to give the cell the toolkit. And so it's like adding new Legos to the Lego kit that's already in the cell. Yeah, pretty much. Yeah. So Elowitz says, one of the biggest challenges in biomedicine is specificity. How do you make a therapeutic that will affect only a particular type of cell? Then how do you respond by modifying that cell in a very specific way? The tasks are challenging for drugs, but biological circuits could excel at them. Protein circuits can be programmed to sense many types of information, process it, respond in various ways. In fact, the reason our cells usually work as well as they do is the incredible power of our natural biological circuitry. And so this is it's kind of naturally based, but it's synthetically engineered. It's a proof of principle, and they haven't actually done any of this in animals yet. It's just the first step in this engineering challenge. Fun stuff for engineers. I've got a couple more stories. How about a story about birds? I like birds.
Yeah, always.
We always love birds. Well, they found another missing link in China. It is a 127 million year old fossil that fills in another gap in the dinosaur to bird evolutionary progression. And historically, birds used to have a long tailbone. They had this very long tailbone and
everything's
feathers.
Everybody fights over who gets the tailbone.
The tailbone, right? Yeah. So Archaeopteryx had a very long tailbone, and many of these older dinosaur-like on the way to bird organisms, they had tailbones. But this new bird known as Jinguofortis perplexus doesn't have a really long tailbone. Its tailbone is stubby. However, modern birds have a stubby tailbone that has a fan of tail feathers on it that's used for flight. And this fossil had no fan of flight feathers. So it was pre-fanny tail and post stubby tail. So it has the stubby tailbone, no feathers coming off of it.
Wow, it looks really weird.
So what came first? The tail feathers or the stubby tailbone? Now we know.
Now we know. Stubby tailbone came first. Exactly.
And still has teeth. Frightening.
It did. It had its jaw had small, sharp teeth that was similar to theropods. And still, chickens today may not have teeth, but they have the genes for teeth, and we could possibly We've done it. We've given chicken teeth. Yes, we have. There were remnants of gizzard stones in the fossil, which suggests that Perplexus was a plant eater because, as we said earlier in the show, cellulose in plants is very hard to digest. One of the ways birds deal with that, stones in the gizzard. And its third finger consisted of 2 bones, which is an early example of, which is unlike other early birds. And so it's an early example of a bird species reducing its finger bones.
Fusion.
Fusion of the bones.
I like
Just-
its third finger was 2 bones. Wait, that would be 2 fingers. Wait.
Yeah, it had a unique shoulder. They're fused together to form a scapula choroid, which is all the, if you could imagine your shoulder being all fused together. And they think this helped the birds grow faster while they were developing. Yes.
This part always is fascinating and simultaneously drives me nuts. Is that this is the picture that they looked at this.
Smashed. It's like a smashed fairy.
And these scientists are so good at what they do that they see this mash and they're able to pick out this. Like, it's just the fact that you can even look at that and say, oh, they don't have a lot of tailbones.
And so this bird, this early bird, late dinosaur, looked more bird-like than it did Archaeopteryx-like or dinosaur-like.
It's really fascinating to me that this little smush can give you so much information. It's really cool, actually.
It's pretty amazing. Yeah.
Hmm.
Yes. So it's an interesting new bird in the evolutionary path of birds. And in terms of bird is the word, the biggest bird in the world has been discovered.
Bigger than the elephant bird?
Oh, yes. This bird, the largest bird ever to have existed on the planet, it's known as Vorombe Titan. Vorombe Titan it weighed in at about 1,700 pounds, and it stood up to 9.8 feet tall, or more than 3 meters.
Oh, dear Lord.
Wait, that's
taller than a man.
That's about elephant birdish.
No, elephant birds weigh about 800 pounds.
No, they
Yeah.
were more than that.
No.
And they were the same height.
The height might be similar, but they weighed about 800 to 880 pounds.
Where is this creature found?
So let me tell you, they've been looking at these elephant birds, trying to figure out these.
It's a relative.
It's a relative of the elephant bird.
This is not a whole separate giant bird outcrop.
This is a group of birds that are known to be very large, otherwise known as elephant birds from Madagascar.
On Madagascar.
Yeah, so the elephant birds were up to 880 pounds. They laid eggs that weighed up to 3 pounds. But there is this Vorombe Titan is different. This is a different bird. And the researchers were looking at these Apiornithidae family of elephant birds, and there's 2 genera within called the Apiornis and the Molleronus. And the researcher Hansford, who was involved, he was measuring all these elephant bird bones at museums around the world, and he just thought it was this large bird is going to be one big individual from one species from one of these 2 genera, right? And he started doing a cluster analysis, cladistics, trying to actually figure out how these bones were all related to each other. And they realized that these Vorombe Titans, these Vorombes, they are different, distinct genus from the other birds. So there is a third group of elephant birds, much larger, the Vorombe group, and it was giant. The analysis,
so were they overlapping?
the biggest bones differed so significantly that they had to be completely separate.
So
Yes.
this bigger version, though, is this an ancestral bird? Is this one that was at the same? Because the one that
It was about at this. It was according to the study, they lived about the same time as the other birds, the Apiornis and the Molaronis. They were large fauna on Madagascar, and they had a giant impact on the land.
like a woolly mammoth might if it was allowed to
Exactly.
return to the Arctic.
Exactly.
So
Mhm.
so that's wow. So then the elephant birds, this is Madagascar. This is an island. This is
Mhm.
one of these separate evolutionary events. And that means the elephant bird history for such a megafauna on an island
Yeah.
must have existed a lot longer than I thought.
Yeah.
For them to have
For millions of years. Millions of years. Yeah.
Wow.
Yeah, the researcher Hansford, he says, interestingly, their closest relatives are the kiwi, suggesting that their ancestors arrived in Madagascar by flying there, and then they evolved flightlessness over millions of years.
Why leave? This place has no cats or snakes.
Yeah.
Cool.
I did not think
Right.
it was possible to like these birds you just showed me more, and now I do.
The Varombe Titan. Varombe also meaning big bird in Malagasy.
Oh,
Big
my
bird.
God. I love Malagasy. It's so fun. Varambe Titan. Yes.
Varombe Titan. It's like
Varambe's a good word.
Yeah.
big bird titan.
Wait, does Varambe mean bird or big? See, now I got to get into it.
Big bird. Varombe. Big bird. Big bird. All right. Give me some more stories. Let's finish out this show. Quick.
Goodness. Food of the future? Quinoa. And not just because the hipsters love it. It's because they are a salt-tolerant crop. University of Würzburg found exactly how quinoa does so well in saline soil. The more agriculture we do, more irrigation, saltwater intrusion, more use of pesticides, all these sorts of things often increase the salt content in soils. Yes, Justin.
So apparently the hipster bus passed me by.
Oh,
What are you talking about? What is
quinoa. Quinoa.
What?
It's
I
a
have
little...
no idea. These words are not
Oh.
of any language I can detect. Even
It's a little rice-like grain that it has almost like a nutty flavor, but it's usually a replacement for rice. It's gluten-free. It's very it's high in protein, but it's a grain, so it's all over the place. If you come down to San Francisco, you'll find it everywhere. But
Okay.
anyway,
Come north to Portland. We've got lots of quinoa here.
Really?
Portland, San Francisco, we got all the
Jump on the
quinoa.
quinoa train.
Yeah.
So
so
if
good.
here's what happened. I've probably seen it in a supermarket,
Yeah.
but it said gluten-free,
Oh.
and therefore I didn't look at it
Yeah.
again. That's probably what happened. Why I've never encountered this.
It's quite delicious. It's easy to make. You make it just like rice. And as I said, it has a high protein content. So it actually has potential to be really valuable as a crop. As a crop food.
Especially with the ability to be grown in high saline.
Thank you.
environments. That's
Yes.
the key. That's the key
Yes.
point here.
So-
As soils change and climate changes, this could be a boon for
Yes.
farmers.
And it gets even better. So this research, they actually found out how they are able to grow in this saline environment. They sequester salt in hair-like bladder cells. So they have these storage sites, and they have these storage areas build up saline in their tissue, but it's a one-way channel. So this salt builds up and never goes back into the rest of the plant.
So
So you don't have to add salt later.
Right. Yeah.
No
So that's
salt
only if
needed.
you're eating the leaves. So it doesn't get into the actual quinoa at all because it's a one-way channel. So they sequester the salt. It prevents backflow of sodium, no salt leakage into the leaves. Even when there's the stored sodium reaches high or critically high levels. It does not come back. So 2 things here. One, this is a great crop as salt becomes more of an issue in soil. But the
sea level
second,
rise, it's saturation. It's
yeah.
And let's wrap it
Drought,
up, you
all that
guys.
stuff, yes.
This
But
show's
the-
been going. Let's
yes,
wrap it up.
The second
Oh my
part
God.
is that now that they know the methodology of how this crop is doing it, they think they might be able to potentially get other crops to do this.
Nice. And Justin, your last story,
That was
please.
it. I'm done. I'm out.
You're done? Are you sure?
Yeah.
Nothing about DEET?
No.
Nope. All right, let's wrap it up. We're done. We are finishing this show now, everybody. It's been a long night. I'm a tired person. I hope that you all have enjoyed this show very much. Thank you to everyone out there. Thank you to you who are listening, you who are watching. Thank you for joining us tonight. Thank you to our chat rooms for chatting and commenting. It's wonderful to have you here when we're doing the show live. Thank you to Identity 4 for recording the show. Thank you to Fada for helping out with show notes and social media. And I would love to take this moment to say thank you to our Patreon sponsors. Thank you to Paul Disney, G. Burton Lattimore, Richard Onomus, Ed Dyer, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Andy Gross, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, John Ratnaswamy, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard, Brian Condren, Dave Neighbor, Aiden Jeff, Dave Wilkinson, Ben Bignell, Richard Porter, Jacqueline Boyster, Noodles, Kevin Reardon, Christoph Zuknurek, Noah Zemke, Ashish Panth, Ulysses Adkins, Sarah Chavez, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Slazewski, Jim Drapeau, Greg Riley, Sean Lamb, Ben Rothig, Steve Letherman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, EO, Mark, Tyrone Fong, and Keith Corsell. Thank you to you all for your support on Patreon. And for any of you right now who might be interested in how the Patreon thing works, you can find information at patreon.com/thisweekinscience, or you can just go directly to twis.org and click on the Patreon link. Remember that you can help us out also by telling people about TWIS. Share your love for TWIS if you are so inclined. On next week's show, we will be back. And we're going to be talking about plastics in the ocean, microplastics. We've got an interview next week with a Oregon State University researcher who is going to be speaking with us about her research into microplastics in the environment. Also, our show starts Wednesdays, 8 p.m. Pacific time. That's right. Every Wednesday, 8 p.m. Pacific time. You can come join us at twis.org/live. You can join our chat room there. There's also our YouTube channel. Look for This Week in Science on YouTube. If you can't make it, though, our past episodes will be in these places, twis.org and on YouTube for perpetuity, in posterity, forever, ever. And you can go check them out there.
Keep the conversation going on forever. TWIS is also available as a podcast. If you want to find it, you can go to your iTunes, Google This Week in Science, should pop right up. Also, if you have a mobile type device, you can look for This Week in Science, or no, TWIS, the number 4 Droid app in the Android marketplace, or simply This Week in Science and anything Apple Marketplace.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org, where you can make comments and start conversations with the hosts or other listeners while you're there.
Yes, or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, hey, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just get understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gambiae. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 This Week in 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 Science.
Oh, it's going to be a very short after show. I have
Me.
oh yeah, I have exhaustion going on here. And I have a very long workday, fun workday ahead of me tomorrow, but a very long workday ahead of me, and I would like to be rested for it. I think.
What is this rested you speak of?
I was talking with my brother, and he's like, "Yeah, you take the Mountain Dew, and you drink the code red when you're doing that, and then you drink that up." I'm like, I don't even know what you're talking about.
You have a brother?
I have a brother. I have a brother.
I didn't know that. Does he listen?
Brother.
Hi, Kiki's brother.
I don't know. I think he avoids listening. He's like, my strange sister.
Is he older or younger?
Younger.
Younger.
Yeah. My sister with all of her science.
Is that your only sibling?
It is.
Oh.
He is my only sibling. Yes. Hey, fecal lord with the fly in your shot glass. I would like you to pour a shot without a fly in it and tell me if you can smell the difference. I would like to know if it is the same as the master wine tasters.
Yes.
Who can tell the difference between glasses of wine with and without flies. My favorite studies like that. It just it cracks me up. I love it. I love it. Yes. Oh my gosh. And yes, I picked on my brother when I was younger. I was a typical older sister. With typical. You can already won't be able to smell the difference. All right, fine. Just drink it anyway then. Who cares?
I love that we're going on this emotional journey with Fecal Lord right now. I really feel close to
He's like, I'm gonna drink it anyway. Yes, it's alcohol, right?
This is quite the narrative that we're being supplied right now. I really feel
in the chat room
like I'm traveling
Yeah,
with
the
this
emotion-
person.
the emotional journey for sure.
Yeah.
Yeah,
Good work, Fecal Lord.
I'm gonna drink it. We gotta do it. Drinking it. I mean, you would cringe taking a shot of cheap whiskey as it is. So
Yeah.
questions.
Yeah, couldn't
So
taste
many
it.
questions.
Yeah, how big was this fly? That's the other question. Like, was it a little fruit fly? That's like not a big deal. If it was like a big horse fly, I don't know if I could drink that shot. Even if I picked it out. I think it'd be too much for me personally. Too big. The ratio of fly to
Apparently, it did not taste any different. Thanks for joining us, Lightning Rod. Did I do science to my younger brother? Asks Jeremy. Probably you could call it that. Oh my goodness.
let me get my calipers and measure it.
There you go.
That's how you can tell a
Scientific.
scientist.
That's right. Scientific
That's like-
method in hand. There we go.
That was like when I did some prepping of skulls a few weeks ago, and I was like, "Do we have goggles? No, I'll grab mine."
Yes.
Still have my lab goggles just
I have
around the
m-
corner here.
I have many goggles.
Yeah.
I love my goggles.
Well, mine are starting to get kind of scratched on the lenses, which isn't good because you need to be able to see really well.
Yeah.
Like a big bummer when the goggles get cracked.
So this week I am doing more.
I come back and everybody's yawning.
I know I'm very tired. I must be fresh for my video shoot tomorrow.
Oh, good gosh. Okay, but in that case,
Yeah,
say
I am
goodnight, Blair.
doing. I'm
No, she's
doing-
gonna tell us first.
I was going to tell you what I'm doing. Am I allowed to
Oh.
tell you what I'm doing?
Yeah. I just thought you were like it was a secret.
I don't think it's a secret. They haven't told me. It's not a secret. I've been on this program before. What on Earth? On the Science Channel.
Nice.
I have been invited back to be part of the next season of programming. I'm not going to tell you what stories are in the program.
Oh, why? What are they?
The stories I'm talking about are very fun and interesting. Really fun stuff. Let me tell you that. I have fun. It was
If you listen to TWIS, you probably already heard the story, right? Or is it a different
some of the science, maybe, but not the stories
Ah.
necessarily. I mean, not the way it's presented in What on Earth. But yeah, some of the
I'm
stories
intrigued.
you may have heard,
I'm intrigued.
or the science behind them you may have
But
heard.
now
in
with
the Secret
production
Science.
value and time to flesh out the story, it's probably even more.
Yeah.
Hmm.
Yeah. So very fun. I had an 9-hour video shoot
Wow.
day yesterday.
Holy smokes!
And I had a prep for TWIS and like a little bit of a day off today, and then I have another 9-hour day ahead of me tomorrow. So yeah, I've got a long day. We've got many stories to get through. The thing that's great is they like how I present stuff, so they give me a whole bunch of stories to describe and talk about and potentially be edited and be part of on the show, which is great.
Well,
but it also means it's a lot of stories to get through.
it
It's a
is.
lot of
Yeah.
talking and being on,
Yes.
on all day.
However,
Yeah.
as has been brought up in the past, there is not a human on the planet now or who has ever existed who has brought more science stories to the public than yourself.
I don't know if that's true,
I'm
but
pretty
okay.
damn-
I
we
like
have
your
done-
I like your optimism there.
No, it's not optimism. It's partly it's attrition. We've done this for I've done this for 13 years. You had an extra number of years when it was just radio show. Beyond that, what are we at? Are we at from the actual inception of TWIS? We're starting to get to the 20-year anniversary.
I think we're pretty close because
Yeah.
didn't
99ish.
I feel
Yeah,
like
we are very close.
So-
2
so-
years
so-
away-ish.
And within that,
Yeah.
every episode you tend to bring more stories than the rest of us. And no, this is a superpower. And there was a period when we would do the show with 3 to 4 stories each,
Yeah.
and then you would do a roundup rundown of the stuff we didn't cover. So just by sheer number of stories that you have put out into the public arena,
It's a few.
I say there could possibly be none who have surpassed you in raw number of stories brought. So the fact that they go, wow, we need to give her more airtime because she's really good at communicating the science is not something that anybody should be surprised about.
Right.
But it does
Not
mean that you've
surprised.
got to be on your game tomorrow.
I do. I will be on my game tomorrow. I'm going to go to bed. And let's see. I'm doing a Friday Twitch stream, regular Twitch stream Fridays, to talk about science over on Twitch, Dr. Kiki channel. I'm still working out what we're going to do with the This Week in Science Twitch channel. We're going to do some broadcasting there, but yeah, so I'm starting to do that. And this Friday, I'm going to also be heading to a Oregon State University ship. They've going to have a ship in harbor here in Portland, and I'm going to go talk to scientists who use this research vessel to to study the oceans. Yeah, there's one researcher who I believe launches drones from this vessel to study whale poop from above.
yes.
Yes, I'm going to hunt this person down on Friday morning.
I've heard about the whale poop vessel.
The whale poop vessel. It is a vessel for finding the whale poop.
Mm-hmm.
I severely hope that you come back just only talking like a salty sailor.
Salty sailor. Oh my god, it would be a curse word every other word. Arr. Arr, matey. Oh my gosh. Yes. Okay. I think that's the news for what's going on. There will be a link showing up sometime in the next week on the website to buy the calendars, pre-order
you
the
go.
calendars. I have
That
images.
time.
I'm going to get on it. I will send out social media notes when the link is live. Right now it is not. I will let you all know. But yes, calendars will be available in the next week for pre-order. Is there anything else? We are at episode 689, which means we are 11 episodes away from 700.
Close.
Technically-ish, right? So, yeah. Oh, and I know that we said we weren't going to do a Halloween episode, but Blair said that it would be okay to do a Halloween episode because her Halloween is like Saturday and not Wednesday.
Yeah.
So Blair, maybe would you be okay with running the show if I got you a guest host?
Yeah, that can work.
Or if you want to bring in a guest host, do you want to bring in other zoo or science friends?
That is a lot of pressure to do live on the air, Kiki. She's like,
Thank you.
there's a lot of people allowed to bring, but I'm going to overrule it and be like, no, we need a professional. This is a professional. I'm sorry, you can't.
Let me think about it. I'll think about it. We
Okay,
have some time.
Think about it. Think
Yeah.
about it. Justin and I are going to be doing trick-or-treating type stuff.
I figured.
We
Yeah.
won't be
yeah.
there. But if you would like to do a show, I mean,
Okay.
that'll be one more episode, and that'll make the 700 episode more likely to be like, right, like a birthday party for you and Justin.
Oh, yeah. So, 90. Yeah, it would be right on our birthdays.
Mm-hmm.
It would be on my actual birthday.
Mm-hmm.
Hmm.
Yeah. So we could either have 700 as a birthday party or not,
Mhm.
depending on what we decide to
Okay.
do with these holidays.
Okay, I will
Think on it.
ponder. I will ponder.
Yeah, the hippo interview. That's gonna be fun.
Yeah.
Gonna be a hippo lot of science.
Yeah, I'm trying to think.
Well,
know.
don't try too hard. It's
Ah.
late. Say
Yeah.
goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin.
Good night,
Kiki.
Kiki.
Good night, everybody. I'm really tired. Okay, thanks for watching. We'll see you next week. I'll see you on Twitch Friday if you join me there. Have a great week. Happy science to all, and to all, a good night.